The Ethics of Brain Organoids and Chimeras by Rovi Campson. Advancements in the field of biology have led to the lab production of mini organs, which mimic the structure and functions of organs in our own body, such as intestines, heart, and even brains. These mini organs are more formally known as organoids. Organoids are often made of pluripotent stem cells. Pluripotent meaning it has the ability to develop into many types of cells. Scientists use growth factors and different types of culture media to influence the type of organoid produced. Organoids are revolutionary in modeling diseases and precision medicine, where treatments are tailored for your specific genetic profile. In the future, organoid technology may hopefully advance to creating transplantable organs in the lab. A company called FinalSpark created a program called Neuro Platform. It uses the computational power of brain organoids to create simulations such as the flight of a virtual butterfly. This butterfly is controlled by 16 brain organoids on a multi-electrode array, each made of 10,000 neurons. The organoids receive electrical and chemical signals that share information about the virtual world, including detection of targets placed by a user. They process the environmental information and move the butterfly towards the target. FinalSpark is quoted as saying,
"It's as if the brain itself is living within this simulated world."
Although the applications of organoids are very promising, there are many ethical implications surrounding the creation and use of brain types specifically. There's fears that, in the future, brain organoids containing the right structures and environment could potentially develop its own consciousness. Currently, there's a lack of standardized tests for consciousness, which could lead to situations where brain organoids develop some type of sentience without researchers being fully aware of it. A study conducted by Trujillo et al. In 2019 trained a regression model on EEG data from preterm human babies and applied it to organoids of varying ages. The regression model was able to accurately predict the ages of brain organoids older than 25 weeks, meaning that the neural activity of brain organoids were analogous to that of preterm babies. The similarity outlines the potential for further cognitive developments as organoid technology progresses. If brain organoids evolve to the point where they can perceive pain and are self-aware with their own interests, then many authors argue that they must be assigned a moral status, which involves considerations around its rights and welfare in research. Human brain organoids have also been used to study its regenerative properties as transplants in non-human species, creating organisms called chimeras. In 2023, a study by Chimeras et al. Implanted human brain organoids into the visual cortex of rats and found that it increased their response time to a light stimulus. The figure on the left represents normal rats, and the one on the right represents organoid-grafted rats. The dots in each row represent spikes of neural activity, and the yellow line signals a light stimulus. Comparison between the figures show that the organoid-grafted rats displayed longer periods of neural activity after exposure to the light stimulus. The authors hypothesized the difference was due to increased neural connections between the human organoids and the rat brains. Ethical concerns arise from the possibility of creating human-like consciousness in these chimeras. The humanization of other species would lead to a morally ambiguous state. If they develop human cognitive traits, such as self-awareness and complex emotions, then would their moral status rise to the same level as humans? It's important to note that current brain organoids are not capable of sentience. They are much simpler than our brains and do not have the capability to interact with the social environment, which authors argue is required to develop human-like consciousness. But who knows, maybe in the future, brain organoids would be able to think as humans do.